Hi, this is Rethik Sir, your biology SME. Today we will be discussing about the anatomy of dicotyledons and monocotyledons plants, especially on the root system. See, what happens is whenever we are focusing to study on specific part or specific structure, anatomy, that is the transfer section of those specific, that too especially in the initial growth, that is the matured zone of these organisms or the matured zone of the organs. If I'm subjecting to plants, the matured zone of those organs, such as it could be their leaf, stem and root plant. So taking the transfer section and studying that will give much information regarding the structural arrangement of the tissues and its respective functions. So in this video lecture, we will be discussing about the root system in both the plants, that is dicotyledons and monocotyledons. Clear? So before getting into the video, one very important announcement is today and tomorrow will be the last day for the Elite subscription, and as well as since it's August 15th today, you have an offer, that is you have offer for both Elite plan and Brahmastra test series as well as PYQ book. Okay, so make use of it, and the information is being pinned in the message box as well as it is been available in the description also, so do check it. Coming to the dicotyledons root, so the outermost layer is actually called as epidermis. So whatever the outermost layer that we see, right? So they are always called as what? Epidermis. So many of the cells of this epiderma protrude in the form of unicellular root hair. Protrude is nothing but they are stick on. That means, can you observe here that present as stick on the unicellular root hair? So these are the root hairs. So they are just like that being sticked on in the form of unicellular root hairs. Okay. Then the cortex consists of several layers of thin-walled parenchyma cells with intercellular space. So this is the cortex here. So cortex, you can see there are several layers of thin-walled parenchyma cells. So these are the parenchyma cells. So there are several layers which are thin-walled parenchyma cells with, can you see a spacing in between? So intercellular space in between those parenchyma cells. So that is called as cortex. Then we will be able to see the next part, that is endodermis. The innermost layer, endodermis is nothing but the innermost layer of this cortex. After cortex, again, if we go deeper, then the innermost layer of the cortex is actually what? Endodermis. It comprises a single layer of barrier shaped cells. So only single layer of barrier shaped cells like this. Okay, single layer of barrel shaped cells without any intercellular spacing. That is, you will see the cells, they are being packed close to each other such a way that there is no intercellular space. There is no space between the cells. So that is what they are trying to say. Then after to that, we have a tangential as well as radial walls of the endodermal cells have a deposition of water impermeable. That is nothing but here what you can see is again after this endodermis. Okay, so you will see that they are tangential as well as radial wall can be observed, okay, of this endodermal cells. So what happens is they are water impermeable. That means they actually don't allow the water. And they are actually made up of what? Wax material in the form of Casparian stripes. Okay, in the form of what? Casparian stripes. Then next to this endodermis, next to this endodermis, there is again a few layer of thick walled parenchymatous cell. Okay, so they are actually referred to as pericycle. Okay, they are referred to as what? Pericycle. Here, what happens is initially, it normally initiation of lateral roots and vascular cambium. So here what happens, you will actually see here the lateral roots and vascular cambium during the secondary growth takes place in these cells. That means what happens? So we have this cambium, right? So do you remember the structure here in the vascular tissues? So what there was in between the xylem and phloem, so we had a structure called cambium, right? So what is the major? So in whichever the vascular tissue where it has the ability to produce or to become a secondary growth takes place, right? So there cambium is present, and wherever the secondary growth is not, it doesn't happen, there the cambium is absent. So in dicotyledons, what happens? The cambium is present. That is what they are trying to say. So this was all regarding dicotyledons. And another very important thing is the pith is small. After this region, you will see the pith region. So pith region, as you can see here, these are the pith region. Okay. So this is the pith region, which is very small. And here this parenchymatous cells, okay, which lean between. So where they are present, they are present between the xylem and phloem. So they are the xylem and it is the phloem region. Okay, yeah, if they are the xylem and if they are the phloem, right? So what happens between them, this pith region is present and it is very small. So that the parenchyma cell which lines between this xylem and phloem are actually called as what? Conjugative tissues. So whatever the cells which lie between this xylem and the phloem is actually called as conjugative tissues. This is another very important point. Now there are usually 2 to 4 xylem and phloem patches. Okay. Later, what happens, as I told you, this cambium, cambium ring, that is like this. Later, this cambium ring, right, develops between this xylem as well as the phloem. Getting? So what happens? Later, this cambium ring is going to develop between this phloem as well as xylem. And all tissues on the inner side of this endodermis, that is, inside this endodermis, whatever the tissues you are finding, right? So, such as pericarp, you can see. Then we have vascular bundles. Then we can also see what? Pericycle, vascular bundle, pith region. So these all, it actually constituents together the stele, stele. Okay. So this was all regarding what? Dicotyledons root. So whenever you take a transfer section of the dicotyledons root, so what are all the things that you are going to see? First thing is the epidermis you are going to see. And this epidermis, they are the outermost layer. Okay. And you will also see or observe a root hair. After that, we have the cortex, and in the cortex, it consists of walled uh, parenchymatous cells, and in between there is an interspace, okay, with intercellular space between. After that, we have endodermis, which is single layered and there is no space, intercellular space in between. Then again, the pericycle, which follows the endodermis, and after to the pericycle, we have pith region, so which is very smaller, and the parenchymatous cells, which actually lie between this xylem and phloem, they are actually called as what? Conjugative tissues. And lastly, you will also see the development of the cambrium in between the xylem and phloem later on, okay, so that development actually takes place later on, and all tissues, okay, which is present inside this endodermis, such as pericycle or vascular bundles, then pith, they all constitute what? Steel. Clear about this dicotyledon's root? So can we move to the next slide then? So what would be the next then? If it was dicotyledon's root, then the next will be obviously the monocotyledon's root. So was it very simple? Was it very simple? Only thing I was supposed to remember is this figure, right? The figure and the labeling. So if we are aware of this labeling part and aware of this where exactly what comes after what next, so then it becomes easy to remember its function also. The next one is monocotyledon's root. In monocotyledon's root, now we can easily differentiate the structure. Structure is somewhat different, right? Structure is somewhat different. So, in the monocotyledon's roots, what happens? It has epidermis, then it has same. It has the epidermis, it has the cortex, it has endodermis, then pericycle. It also has vascular bundles and pith. Everything is same. But as compared to this dicot root, okay, when we compare this monocot root with the dicot root, as compared to the dicot root, when we compare it with the dicot root, which this monocot root, okay, there have few xylem bundles. That means here you can actually see some more xylem bundles can be absorbed. Fewer xylem bundles, okay. So what happens here? We had many xylem bundles such as protoxylem, metaxylem. But when we are seeing on the monocot root systems, okay, what we are being able to see only fewer xylem bundles, say like protoxylem, okay. This is one major difference. There are usually more than 6 xylem bundles in the monocot roots. Then pith is larger and well developed. So here there the pith was actually small, but here the pith region is smaller or larger. Here the pith region is actually larger and they're well developed. So in this monocot roots, what happened? They do not undergo secondary growth and there is an absence of cambrium. So you won't see any cambrium in between the xylem and phloem. Therefore, what happens? They do not undergo any secondary growth. So this is the major difference between the monocot and dicot. So till that is same. That is just like the dicot, you have epidermis, cortex, endodermis, pericycle, vascular bundles, pith, everything is there. But only thing differences is here as compared to the dicot which has fewer xylem bundles, there are usually more than 6 xylem bundles in this monocot roots. Okay. Then coming to the pith is larger and well developed. This monocotyledons root or monocot roots, they do not undergo what? Secondary growth. Clear to you? So is it really clear? The major difference between the monocot and dicot roots. Fine. So this was all about what? In generally, when anatomy of dicotyledons and monocotyledons root system. So there comes another video lecture. I'll be covering about plant system. And now let us detailedly discuss few questions on this particular topic. Okay. Try to solve some PYQs, that is previous year questions or the questions which was asked previous year. Okay. I'll try to analyze how the questions were asked and in what way we need to deal with those questions. So this question has actually been taken by our NTA PYQ book, and the question was asked in previous year, that is 2024 of NEET. So the transfer section of monocot root shows the following internal tissue organization. Arrange them in a correct sequence starting from periphery to central. So when I'm talking about something, I have to understand the diagram here. Okay. So what they have given now, the monocot root showing the following internal tissue organization. So very first thing I need to understand is the figure of the monocot root. So let us go back. Yeah. So this is what monocot root. So in the monocot root, what is the very first that came? Epidermis. The second one was cortex. Then we had endodermis. Then pericycle. Then we had peth. Right? So in this way, it has gone. So now let us see the question now. So what will be your very first? Epidermis. What will be your second now? Cortex. What will be your third then? Endodermis. Very good. So what will be your fourth? Pericycle. Yes, good. So the last one will be obviously the pith region. So which of the following is actually showing the correct option now? So C, then we had E, C, E, C and E. Okay. Next one was endodermis. So this is yeah. The four. Then we had the fourth one, that is the D. Then B is pith region. So option C is the correct answer, and let us see. Yes, option C is the correct answer, and this is how the explanation suits. We'll go to the next question. Okay. And this question was also being asked in 2024, and it is directly from NCRT line, that is statement based question. So what the statement is now, the statement one is that the parenchyma is leaving, but Kulankaima is dead tissue. That is what the first statement is saying. That is, the parenchyma is leaving, but Kulankaima is dead tissues. So what the statement two is, what the statement two they have given, the gymnosperms lack xylem vessels, but presence of xylem vessels in the characteristics of angiosperms. In the light of the above statement, so this is something like what? It also comes under plant kingdom question also. So that is linked with this present chapter also. In that way also we can see. See, one thing we know, the gymnosperms, yes, of course, they lack the xylem vessels, but angiosperms are nothing but the one which were able to bear the flower. So they are flower plants. So they had this characters was present, that is presence of xylem character was xylem vessels was there is there in the angiosperm. So with the above two statement, which of the following option is correct? So statement 2 is also correct. So with this, if I have to consider the statement 1, with this, if statement 2 is correct, so if I need to consider statement 1 with the statement 2, then statement 1 is wrong because both parenchyma and collenchyma are leaving. So where collenchyma is dead is wrong. So according to that, the statement, according to that, the statement 1 is false, but the statement second is true. Let us see the answer. Yes. The parenchyma is living, but collenchyma is dead. Tissues is fall, and the statement 2 is right. Reason they have given here, the parenchyma tissue is living as well in the collenchyma, which is living. So both of these are examples of what? Simple tissues. So whereas the sclerenchyma is a type of simple tissue which are dead, not living, important for the structural support. Okay. So sclerenchyma, parenchyma and collenchyma are living. What you will be seeing, there is another thing that is called sclerenchyma cells. They are actually a type of simple tissues which are dead. That means they are not living, but still they are important for the structural support. So therefore, both parenchyma and collenchyma are living tissues. Okay. So that is what they are saying. And same thing to the statement 2, where gymnosperms typically they do not possess the yes, possess vessels elements in their xylem, in contrast to the flowering plant where there is both vessels and tracheids. So here what happens, this vessel elements that are xylem, phloem are predominant in the angiosperms, that is flower bearing plants. So while they are not, or they are not notable, or their absence in where? Gymnosperms. So at the end, you can also see which chapter, which topic, and what kind of statement it is, and from which page number the question and explanation has been taken from. So having a PYQ, especially NTA PYQ, will actually boost your confidence to know how the preparation is, how the question pattern of the NTA is. So yes, we have a sale where presently for today and tomorrow we have offers. You can go to the description, find the suitable offers, and you can contact the given WhatsApp number. Thank you and all the best for your preparation. And in the next video chapter, we will be discussing about the monocot stem and dicot stem transfer section. Thank you one and all.